Good morning. Many thanks to PacBio and the organizers for inviting me here to speak today. For the next 25 minutes, we're going to talk to you about ALS. I'm going to start by talking to you about how Target ALS, a nonprofit organization, is leveraging HiFi sequencing to uncover the missing heritability in ALS, and then I'll turn it over to my colleague, Evan, who's going to specifically talk about the C9orf72 repeat expansion, the most common genetic cause of ALS. And at the end, I hope you'll have a better understanding of how a nonprofit organization is using a unique approach to power ALS research. So if you're unfamiliar with ALS, ALS stands for amyotrophic lateral sclerosis, which refers to the progressive degeneration of motor neurons in the brain and spinal cord that eventually leads to the loss of mobility, the loss of speech, the inability for these patients to communicate with their loved ones, and ultimately loss of their ability to breathe independently. This disease is always fatal, and there is no known cure. ALS is a complex, heterogeneous disease. Both the underlying biology and the clinical manifestation can differ across patients. Dan Doctoroff, our founder, is a good example of this. His father and his uncle both had ALS. They both tested for C9, a hexanucleotide repeat expansion in the C9orf72 gene. So when Dan thought about his diagnostic journey, he decided to get tested, and he was negative. So when he became symptomatic in 2021, it was confusing and surprising to find that he had ALS. And today, he still has no understanding of what causes his disease, and this is true not just for Dan, but for 90% of people living with ALS, even for some of those who have a strong family history. So the good news is that we've learned a lot from the genetic research in ALS over the years. So I want to highlight 3 important points on this slide. The first is if you look at the timeline on the left, you can see that in 1993, the first mutation discovered to cause ALS was detected in the SOD1 gene. Since then, at least 30 to 40 additional rare and common genetic variants have been identified. Now, converging evidence suggests that there are many more variants to be discovered. Second, what you can appreciate from the figure is that the frequency of these discoveries increases in the 2000s, and this was very much connected to the improvements in sequencing methods during that time. Third, we now know that the FDA-approved drug, tofersen, which targets the SOD1 mutation, provides meaningful benefit to patients. So taken together, we believe that uncovering more genetic risk factors leads to better diagnostics and to the next wave of effective therapeutics. So Target ALS is tackling missing heritability in ALS by combining large-scale data generation with HiFi sequencing technology to capture the genetic variation missed by earlier approaches. We're building the largest HiFi sequencing collection for ALS, and the data are made easily accessible through our data engine, a rich data resource, web-based platform containing thousands of clinical, demographic, and multi-omic data points. The data engine is run on DNA Stack's Omics AI platform, making it possible to connect our data sets with HiFi-Solve's broader rare disease data collections, and enabling AI-powered discoveries across these distributed data sets without moving the data. So why did we choose to invest in HiFi sequencing? As many of you in the audience already know, traditional short-read methods struggle with detecting repeat expansions, structural variants, complex genomic regions. And so it was really important to us to apply long-read to be able to resolve those repeat expansions in C9, allowing us to more accurately measure the repeat length and to understand which allele carries the expanded repeats. And we also want to be able to detect previously hidden structural variants, one of the biggest suspected sources of the missing heritability. So Target ALS funds clinical research on a global scale. Here I'm going to talk to you about 2 different large clinical initiatives. AGRI, our new ALS Global Research Initiative, is comprised of 2 study arms. A large multi-site natural history study designed to collect longitudinal clinical, digital, and multi-omic data sets. And you can see the clinical sites that are indicated on the map here on the slide. And to complement that approach, to be more inclusive, we wanted to run a second study where we're going out into the community to collect a one-time blood sample for DNA sequencing. So together, we have 420 people enrolled in the study, and we're working towards a goal of generating 6,000 long-read whole genomes over the next few years. I'm excited to say that at least 30% of AGRI participants come from diverse ethnic, genetic, and demographic backgrounds, surpassing the diversity captured in other genetic studies. The second initiative shown on the right is that we've been generating a really robust postmortem tissue repository, and every case is collected at 6 different institutions by expert neuropathologists and clinicians to generate deep clinical multi-omic phenotyping from the tissue. We have 600 cases that includes ALS, FTD, non-neurological controls, and at least 62 C9 cases. So together, these clinical data sets are really powerful and provide the power to accelerate discovery at scale. So just a little bit about the methods that we're using for sequencing and the quality of the data we're generating. Here on the left, you can see the methods that we're using, whether it's enrolling for our AGRI clinical study or whether people at end of life are generously willing to donate their tissue to our postmortem tissue core. They are all consenting to share their samples and their data, genetic data for research purposes. The samples are collected by clinicians and trained healthcare workers. They're stored at -80 and then shipped to our partners at Somagen to extract high molecular weight DNA from either whole blood or frozen tissue. Somagen runs our samples using PacBio's Revio with SMRT Cell, allowing for HiFi sequencing at scale. We conduct variant calling using the standard workflows developed by Google and PacBio. So you can see from the QC metrics on the right part of the slide that we've sequenced close to 300 long reads to date. The average read quality is high. Our Q28 metrics show that we're getting 99% accuracy on our reads. The average read length is about 15 kb. We're achieving about 20 times coverage of the genome, and at least 90% of the samples we submit pass our criteria for sequencing. So altogether, the throughput and the quality of the sequencing is high, and these data are actively being shared with more than 500 researchers worldwide through the data engine. Next, I want to share with you just some of the preliminary findings coming from these clinical studies. So when starting to analyze the data from AGRI, we noticed 2 interesting findings. So if you look at the figure on the left, you can see that the whole genome sequencing data is picking up increased percent of new variants compared to catalogs like GnomeAD. So we're seeing a 6% increase in the variants we're detecting in our study in people of European descent. But interestingly, we're seeing a 16% increase in the variants that we're picking up in people of non-European descent. So this is telling us that we're really doing a good job of getting the diversity that we had intended to do, and that we're providing a more accurate assessment of the variants across the ALS population. So on the right-hand side of the figure, I'm showing you a Venn diagram that shows that HiFi sequencing is detecting many rare structural variants in healthy individuals, some of which are shared with ALS patients. But then you can see in the blue circle that many of these variants are unique to ALS. So we're encouraged that with an even larger data set, some of these unique variants may become validated risk factors for ALS. So as I mentioned earlier, we also wanted to be able to resolve the molecular features of the C9orf72 repeat expansion. So I'm just showing you a snapshot of some of the data from our study. If you look at the table on the left, what we did was first to compare our long read data to our short read data. So the third column to the second column across 10 subjects. The top 5 subjects are C9 positive, and the bottom 5 subjects are C9 negative. But interestingly, we can see that the long read is detecting significantly longer repeats than the short read. So in particular, you can see a change in about 2 to 5-fold. So this is a big difference using the HiFi sequencing. In the figure on the right, I'm showing you an example of the range of the repeats that you can have across 20 different cases. So these repeats can range from across individuals from hundreds of repeats to thousands of repeats. And in addition, if you look at the methylation, which is depicted in the yellow gradient moving to red, the more red that the sample is, the higher the methylation level is. So now researchers who have access to these data can go back into our multimodal data sets and measure the C9 transcript. They can determine if it's downregulated, and they can look at all of the other metadata that's associated with these samples to help determine how C9 repeats may lead to motor neuron degeneration. And so HiFi sequencing is really revealing some of the important molecular details that we need to understand this disease. So in conclusion, Target ALS is catalyzing the next wave of genetic discoveries by generating the largest collection of HiFi sequencing in ALS. We're sharing the data out openly through the data engine to accelerate research, and HiFi sequencing is detecting novel genetic variants in our study and the key molecular features of the C9 repeat expansion. So before I turn it over to Evan, I just want to take a minute to say thank you to the people and the patients who enroll in our studies, our global natural history study investigators, the Target ALS cores, Somagen, DNA Stack, and of course, PacBio. Thank you.
Hi, everyone. My name is Evan Udine. I'm a postdoctoral fellow in Marco van Blitterswijk's lab at the Mayo Clinic in Jacksonville, Florida. First of all, I want to say thank you to the PacBio organizers, as well as Amy, for inviting me here to give this presentation. I'm going to be kind of continuing on from Amy's talk, discussing how our lab is using HiFi sequencing to study the C9orf72 repeat expansion, utilizing the Target ALS Data Engine, as well as some data that we've generated in-house. And I did want to highlight that our lab is part of a team that recently was awarded a Target ALS grant to study disease drivers of the C9 repeat expansion. This is led by Dr. Rosa Rademakers in Belgium. This is only part of the graphic for the project, but as a part of this, I did want to mention that we will be generating targeted long-read, whole genome long-read, and bulk kinetics data that will be deposited into the Target ALS Data Engine as well as the project progresses. So to jump in, I really focus on the C9 repeat expansion in my research, which is recognized as the most common genetic cause of ALS and a related disease, frontotemporal dementia, or FTD. And this is a non-coding hexanucleotide repeat expansion of the sequence GGGGCC, where in the general population, we can expect two alleles that range between 2 and 30 copies of this repeat. However, in affected carriers, you generally see one copy of this wild-type allele and one expanded allele, where you have hundreds to thousands of repeats. However, we've noted that not only is the repeat length variable between individuals, but it also varies between tissues and brain regions within individuals. So you can see in the plot on the bottom right, highlighted in green is the cerebellum, whereas in red and blue, you have two cortical regions where the repeat tends to be longer compared to the cerebellum. And now previously, this repeat expansion was discovered in 2011, and for about 10 years, it was estimated to be nearly fully penetrant. And that is that everyone who developed or everyone who had the repeat expansion would go on to develop either ALS, FTD, or a related disease. However, recently estimates of this penetrance are notably lower. And in addition, there were two recent ASO trials that targeted specific C9 transcripts. However, unfortunately, neither trial showed clinical benefit. And so this is kind of some of the questions that have led us to develop this grant with Target ALS, where we're aiming to evaluate if features of the C9 repeat expansion can modify disease penetrance. We aim to look if there's additional modifying factors outside of C9 itself. And we also aim to understand kind of what happened in the clinical trials, if there was sufficient target engagement or the correct mechanism was targeted. So how we traditionally study the repeat expansion, that was primarily using southern blotting, which you can see an example of in the bottom left. And with the southern blot, you can distinguish the wild type allele from the expanded allele, where you see the wild type on the bottom of the gel and the expansion up on the top of the gel. And this is great for estimating repeat length and detecting the presence of the expansion. You can see, though, that the expanded allele tends to be a little bit of a smear pattern. It's not a completely precise measurement, and the ladder that we use to measure it tops off at about 23 kb, though we know now that expansions can actually exceed that length. And people have attempted to use short-read sequencing to study the repeat expansion as well. And tools like expansion hunters, Amy mentioned, are pretty good at detecting the expansion reliably. However, they can't really reassemble it and can't actually provide an accurate measurement of the repeat length. And in terms of methylation, targeted assays have really demonstrated that a nearby promoter can be methylated and has increased methylation compared to affected individuals without the C9 repeat, as well as control subjects. But they've also demonstrated that the expansion itself is methylated too. However, with the existing technologies, there were no quantitative measurements of methylation within the expansion itself. And that really leads us to why we think HiFi sequencing is the right way to study this repeat expansion. As you all know, it's great for covering complex, highly repetitive regions of the genome. Can be used to identify structural variation, as Amy said, is proposed to account for some of the missing heritability in ALS and simultaneously detect DNA modifications. So what I'm going to show for results today is some whole genome sequencing data that we've leveraged from the Target ALS Data Engine, as well as targeted sequencing that we've generated in our own lab. So thus far, we've just begun to utilize the data engine for the whole genome analysis. Currently, this includes just 14 C9 expansion carriers, where right now we've obtained an average coverage of about 21x, so really in line with what Amy showed for the whole cohort that they've analyzed. And you can see that this is relatively consistent between individuals and across chromosomes with some slight variability. So we did first want to see if we can identify the repeat expansion and actually get a size estimate of the repeat expansion in these 14 individuals. And in fact, we can detect the repeat expansion, and we do have fully expansion containing reads in each of the 14 subjects, so that we can get an estimate of the repeat length. And you can see how this looks for one sample on the bottom left here with a plot of IGV, where you can see 2 reads that have kind of fully spanning reads for the expansion. And we also have some split reads that contain a large portion of the expansion as well. However, we can also use Target to more precisely genotype this region, and that results of that are demonstrated for one sample here in the bottom right in this waterfall plot. If you're not familiar with the waterfall plot, it shows each read for one individual on the y-axis, kind of stacked on top of each other, and they're sorted from shortest to longest. And then on the x-axis, you have the read length. And then the sequence is actually colored, so the expected GG-CC repeat motif is colored in blue here. So you can see that there are some slight interruptions to the sequence that tend to still be GC rich. But these plots demonstrate that we can really clearly distinguish the wild type from the expanded allele, and already with just 5 reads, we can see that there's quite a bit of variability even within this one subject for the C9 repeat expansion. And of course, the major use for us of the whole genome sequencing is not just to look at C9, but it also gives us the power to detect and identify disease modifiers outside C9 itself. And so just as an example, this can be done to screen patients for known single nucleotide polymorphisms like that in TMM106B, which I'm not showing here, but is a known protective factor against the development of FTD. It can also be used to screen for known modifiers, structural variants, and repeat expansions like that in ataxin-2, where intermediate repeat expansions have been shown to confer some risk of ALS, especially in individuals with the C9 repeat expansion. And of course, once the data set is kind of fully analyzed and fully generated at a large scale, like Amy demonstrated as planned, we'll be able to discover novel variants of all sorts, SNVs, CNVs, SVs, and repeat expansions that may modify C9 penetrance, but also may associate with disease as a whole. So now I want to switch to what we've been doing in our lab a little bit. And we've really used HiFi sequencing to study the ALS FTD disease spectrum. And we're doing this by generating targeted DNA sequencing, whole genome sequencing, bulk RNA, and single nuclear RNA sequencing across brain regions and in the blood from patients with ALS and FTD. And today I'm just going to focus a couple slides on targeted sequencing and one on bulk RNA sequencing. So the focus of our lab for this has been really using targeted sequencing of C9, and we've recently published 2 studies using the predecessor to PureTarget, NoAMP sequencing, to capture the C9 repeat expansion. And already with NoAMP sequencing, we were really accurately able to measure the repeat length for C9, detect the sequence purity, and also quantify methylation levels of the repeat expansion. And we did this in 2 different regions. One was the cerebellum and one was the blood. And you can see some of the results on the bottom here. So on the bottom left, you see results from the cerebellar study, which demonstrated that shorter repeats actually are associated with survival after disease onset, where shorter repeats are associated with a longer survival time after disease onset. In the middle, I'm just showing one of the results for the methylation, where you can see that when we phase the 2 alleles, we can see that the expanded allele has significantly higher methylation than the wild type allele. And that's not just for the promoter region, which the previous studies had also shown, but this is actually for all CPGs present within and throughout the expansion itself, plus the surrounding region. And more recently, we've focused our efforts on implementing the PureTarget protocol on the Revio. And what I want to note here is that we get a significant improvement in coverage for the C9 repeat expansion. So you can see an example of a waterfall plot on the right here. This has the wild type allele cropped out, so it's just focused on the expansion. And you can see that we had more than 400 reads covering the repeat expansion itself. And what I think can really be appreciated from this plot is the amount of variability and instability you see in C9. So this is a frontal cortex sample that was DNA extracted from postmortem tissue. And you can see that on the smallest end, we have some reads that are about 5,000 base pairs long, up to about 14,000 base pairs. So really substantial variability and something that we're looking to extend into more brain regions and additional patients. And lastly, we've also been generating bulk RNA sequencing. I'm showing here an example of our kind of older Iso-Seq data that was generated on the Sequel 2, but we're also now doing Kinnex on the Revio as well across brain regions. We did do transcriptome-wide analysis, but just to zoom in on what we found for C9, in these 102 individuals, also from frontal cortex tissue, we found 33 unique transcript variants, which at the time this analysis was done was about 25 more than what was present in the Ensemble reference database that we were comparing to. And with these transcripts, we're able to quantify them and compare them across groups, and also annotate them by the splicing events present within those transcripts, predict the open reading frame length, as well as if they're sensitive to nonsense mediated decay. And what we found was that 6 of these transcripts were actually decreased in expansion carriers compared to both controls and affected individuals without the repeat expansion as well. So just to summarize, C9 penetrance estimates appear to be lower than previously thought, which really emphasizes the need to identify disease-modifying factors. And we really think that HiFi sequencing is the most reliable way to detect and characterize this repeat expansion. In addition, we think the genome-wide and targeted approaches are kind of complementary methods to study the biology of the repeat expansion, and the Target ALS Data Engine is providing us with a large-scale resource that's useful for genetic modifier discovery for ALS patients, both with and without the C9 repeat expansion. So with that, I'd just like to say thanks to my lab, all of our collaborators, the funding sources, and of course the donors, patients, and caregivers who make these studies possible. So with that, I think we may have a couple minutes for questions remaining. Thanks.